Aggregatibacter aphrophilus, a closer look. General information. Aggregatibacter aphrophilus is a species of bacteria that belongs to the Pasteuracea family. It was previously classified under the genus Haemophilus, and thus its older name was Haemophilus aphrophilus. Natural habitats. Aggregatibacter aphrophilus is a common resident of the human mouth and oropharynx, even in healthy individuals. It's frequently found in dental plaque, particularly between the teeth and in gingival pockets. Clinical significance. Although it's generally part of the normal oral flora, Aggregatibacter aphrophilus is an opportunistic pathogen that can cause infections under certain circumstances. It has been isolated from various sites in the body, including blood, spinal fluid, peritoneum, pleura, wounds, and jaw infections. It's occasionally implicated in serious conditions such as subacute endocarditis, brain abscesses, sinusitis, arthritis, and osteomyelitis. These infections are often triggered by dental procedures that disrupt the oral mucosal barrier, leading to a transient bacteremia where Aggregatibacter aphrophilus enters the bloodstream. Gram stain. Aggregatibacter aphrophilus, formerly known as Haemophilus aphrophilus, presents as gram-negative, short rods under the microscope. These rods typically measure 0.4 to 0.6 micron in width and 1.5 to 1.7 micron in length. Occasionally, filamentous forms of Aggregatibacter aphrophilus can be observed. Aggregatibacter aphrophilus may appear singly, in pairs, or in small, and sometimes large, clumps. A distinctive characteristic of this bacterium is its tendency to exhibit bipolar staining. This unique staining pattern creates an appearance reminiscent of Morse code, with alternating dots and dashes, further aiding in the identification of Aggregatibacter aphrophilus. Culture characteristics of Aggregatibacter aphrophilus. Aggregatibacter aphrophilus is a facultative anaerobic bacterium, meaning it can grow with or without oxygen. However, growth of Aggregatibacter aphrophilus is enhanced in the presence of 5% CO2. On blood agar, BA, Aggregatibacter aphrophilus colonies appear high convex, granular, yellowish, and opaque, reaching a size of 1.0 to 1.5 millimeters within 24 hours. These colonies are non-hemolytic, meaning they do not cause the destruction of red blood cells. Some strains of Aggregatibacter aphrophilus may exhibit V-factor dependence, requiring the presence of this growth factor for successful cultivation. When cultured in broth, Aggregatibacter aphrophilus initially forms characteristic granules that adhere to the bottom and sides of the tube, resembling tiny colonies. In subsequent cultures, this adherence diminishes, leading to a more uniform turbidity throughout the broth. Aggregatibacter aphrophilus does not grow on McConkey agar, a selective medium often used to isolate gram-negative bacteria. However, it does exhibit growth on Brucella blood agar. Characteristics overview of Aggregatibacter aphrophilus. This organism is gram-negative. It presents as tiny bacilli. Aggregatibacter aphrophilus grows in both aerobic and anaerobic conditions. It does not grow on McConkey agar. This bacterium is catalase negative, oxidase negative, and urease negative. Aggregatibacter aphrophilus is non-motile. It is resistant to vancomycin but susceptible to colistin. Poem. Aggregatibacter, once Haemophilus known, in oral realms, its presence shown. From dental plaque, it may arise, an opportunist in disguise. Gram-negative, with rods so small, bipolar staining, stands out from all. On blood agar, colonies thrive. In broth, its granules come alive. No growth on McConkey, it's revealed. Catalase, oxidase, urease concealed. Non-motile, yet it can invade. Vancomycin shield, its strength unfazed. A closer look, this microbe's tale, from harmless dweller to potential assail. In balance kept, it plays its part, but heed its power, with mindful heart.